Our interest in this area stemmed from a study that we did a number of years ago at this point, where we investigated the mechanistic basis for effective immunotherapy. We started in a mouse model of breast cancer, the polyoma middle T model that spontaneously develops breast cancer, and we left mice either untreated or treated them with an effective immunotherapy consisting of tumor-binding antibodies and dendritic cell adjuvants. And at two different time points over the course of the immune response, we collected all of the different tissues that we thought might be involved in this response and analyzed them at the single cell level to understand immune cell composition as well as behavior and functionality using mass cytometry. So just a brief overview of mass cytometry. This is a technology that allows us to measure many proteins at a time in single cells. We start with a single cell suspension, we can exclude dead cells from our analysis using a viability stain, and we can multiplex samples together by attaching barcodes to each individual cell that allow us to delineate which sample a cell came from. So we can pool many samples together, stain them with antibodies conjugated to heavy metal isotopes. So each antibody is tagged with a different metal isotope that has a unique molecular weight. And then we can analyze those cells one at a time. The nebulizer will put these into little droplets. They get carried into an inductively coupled argon plasma that will ionize the cell, break the covalent bonds in the cell to produce an ion cloud. We filter the ion cloud using a quadrupole to get rid of all the low molecular weight carbon, oxygen, nitrogen that comprises the majority of the cell, and enrich this heavy metal ion cloud. And then we can quantify the metal ions on each individual cell using a time-of-flight mass spectrometer. And at the end, we result in a data matrix where each row is an individual cell that was measured and each column is the expression value for a given protein in that individual cell. And we can go about many different ways of analyzing the data using conventional approaches as well as high-dimensional approaches. And so what we observed in this initial data set, asking what happens during effective immunotherapy, is that there was broad immune cell proliferation taking place in the draining lymph node, but not in the tumor microenvironment at the time that the tumors were actively shrinking and being rejected by the immune system. So what's visualized here are dimensionality reduction plots, a method called scaffold maps, where each node in this graph represents a cluster of immune cells that has similar protein expression profiles. And each cluster is colored based on whether it changes in its rate of proliferation by measuring the proliferation marker Ki67. And so on the left side, we see in the tumor microenvironment that there's no significant change in immune cell proliferation rate with effective immunotherapy or ineffective immunotherapy. Whereas on the right side, we see a dramatic increase in proliferation across many immune cell clusters in the lymph node at this same time point. So that really led us to ask the question, are these responses from outside of the tumor important for driving anti-tumor immunity and efficacy? And the way that we went about testing this, or one way that we did, was to block the egress of immune cells from the secondary lymphoid organs, such as the lymph nodes. So we asked if we prevent cells from leaving the lymph nodes at the time that immunotherapy is given, will it still be effective? And we did this using a molecule called FTY720 that blocks the sphingosine-1 phosphate receptor on immune cells, preventing them from leaving the lymph nodes. And what we found is if we blocked egress, we lost the efficacy of this immunotherapy in our model of breast cancer. We then asked, is this more broadly generalizable to other types of immunotherapies? And so we turned to a mouse model that is responsive to checkpoint inhibitors, called MC38. This responds pretty well to anti-PD-1 immunotherapy. And we saw the same result. If we gave FTY720 together with anti-PD-1, we saw that the tumors continued to grow rather than being controlled by the immune system, suggesting that immune cells from outside of the tumor were important contributors to the success of the immunotherapy.